The Lasergene Molecular Biology Suite provides molecular biologists with a comprehensive set of tools for sequence analysis. With the Molecular Biology Suite, you can perform virtual cloning using a variety of cloning methods, including gateway multisite, TA, topo, and restriction cloning. The software also includes automated primer design tools for PCR, sequencing, and hybridization experiments. This suite also allows you to perform multiple alignments of large DNA, RNA, and protein sequences using the Clustal Omega, MAFT, MAUVE, and muscle algorithms. Quickly and easily merge alignments or add sequences to an existing alignment. Then analyze results using a variety of views, including a distance matrix and phylogenetic trees. The suite also offers easy-to-use sequence editing and annotation, including gene discovery functionality for identifying and annotating coding regions. You can also use the Molecular Biology Suite to assemble contigs of Sanger sequences, performing assemblies both de novo or against one or more reference sequences. After assembly, you can view the read alignment, as well as the coverage,